Hi friends, let us talk about genetic engineering today. Genetic engineering is a technique, is a number of technique that involve making changes in the genetic composition of an organism. Genetic engineering is also called recombinant DNA technology because it deals with the construction of recombinant DNA. What is the recombinant DNA? Recombinant DNA is basically combination of two different DNA. So this is a vector DNA, a plasmid DNA, a vector DNA, and when it is recombined with another DNA, say this one, then what results is a recombinant DNA. This is a recombinant DNA. So all those techniques that are involved in making this recombinant DNA will be together called recombinant DNA technology or genetic engineering. Genetic engineering or recombinant DNA technology involves creation of new individuals with those are called genetically modified organisms or genetically modified microorganisms. These are also called transgenic organisms. And the process is also called transgenesis. So the techniques involved in making these genetically modified organisms will be together called genetic engineering also. But it is not that always we need to have a genetically modified organism for the sake of genetic engineering. There are targets. The purposes may be to know the function of the gene, characterization of the gene, sometimes gene editing, all these things which are involved in characterizing and knowing the function of the gene or storing it or making transcript of the gene, and then seeing the function of the transcript and its location in the cell. All these molecular biological techniques can be together called genetic engineering. But in true sense, genetic engineering involves engineering as such with the gene, with the DNA element. When you cut a DNA element and then you ligate it with another vector or DNA element, then it is, in true sense, it is genetic engineering. Otherwise, the whole subject is called the molecular biology. So genetic engineering involves a number of steps. Those steps can be enumerated as follows. The first of all, we need to have insert DNA. Insert DNA, that is this part of the element, this part of the recombinant DNA. You need to have that first, and it is the particular gene of interest that's need to be transferred, gene of interest. So you need to isolate first the gene of interest. Then the next thing you have to select a vector DNA. Vector DNA is a vehicle. It is usually a plasmid or a phage DNA, or it may be a combination of the two like cosmids, phagemids, and many more types of vectors have been designed nowadays. The next thing you need to have is a number of enzymes. The most important among them are restriction endonucleases, by which you make cleavage in the DNA, both the insert DNA and the vector DNA, for the cloning purpose. Then you need to have ligases with which you ligate the two DNA elements, and then you need different types of modifying enzymes that might involve DNA polymerase, RNA polymerase, clinophragments, and then polynucleotide kinase, simple alkaline phosphatase. Many different types of modifying enzymes are required for genetic engineering purpose. That will be discussed in some other class.